Good day, we are the first group and we will be reporting about the field of industrial biotechnology. I am Bianca Belmonte and I will be discussing about what industrial biotechnology is. Industrial biotechnology, also known as white biotechnology, is the modern use and application of biotechnology to sustain and produce chemicals, materials, and fuels from renewable sources using living cells and their enzymes. This field is widely regarded as the third wave of biotechnology, distinct from the first two waves, which are medical or red biotechnology and agricultural or green biotechnology. It features biotechnological processing that uses enzymes and microorganisms or plant or animal cells to make products in a wide range of industrial sectors, including chemicals, pharmaceuticals, food and feed, detergents, pulp and paper, textiles, energy, materials, and polymers. This field has gathered interest globally because it is associated with reduced energy consumption, greenhouse gas emissions, and waste generation, and may enable the paradigm shift from fossil fuel based to bio based production of value added chemicals. The concept of industrial biotechnology is not a modern idea. In fact, in the past, society has used enzymes in yeast to mass produce beer and bread. Moreover, fermentation processes have been employed to create wine. Over time, humanity's knowledge of fermentation increased, enabling the production of cheese, yogurt, vinegar, and other food products. In the 1800s, Louis Pasteur proved that fermentation was the result of microbial activity. Then, in 1928, Sir Alexander Fleming extracted penicillin from mold. In the 1940s, large-scale fermentation techniques were developed to make industrial quantities of this wonder drug. However, after World War II, the biotechnology revolution began, giving rise to modern industrial biotechnology. Since then, industrial biotechnology has produced enzymes for use in our daily lives and the manufacturing sector. Recently, the utility of industrial biotechnology for bioproducts and sustainable reasons has emerged in attempts to eliminate unsustainable production patterns to reduce environmental impacts and improve the quality of life. The industrial biotechnology revolution rides on a series of related developments in three fields of study of detailed information derived from the cell: genomics, proteomics, and bioinformatics. Industrial biotechnology companies apply these studies and many specialized techniques to find and improve nature's enzymes. To summarize, industrial biotechnology is not only a means to make products in multiple industrial sectors using enzymes and microorganisms. It is also an industrial paradigm based on the expectation that renewable plant-derived carbohydrates, lipids, and other compounds can reduce a significant fraction of petroleum and other fossil fuels, which is currently the raw material and energy basis of modern industrial societies. Along with the mass production of valuable products, its new objective is to develop biotechnology approaches that will yield green industrial processes that are cost-effective and sustainable.
In this section will be the discussion of the people who are involved and who benefited in industrial biotechnology. Around 6000 BC, industrial biotechnology started to emerge when product improvements by certain people were integrated. The people behind industrial biotechnology are the following. First, the Neolithic culture and Babylonians. In 6000 BC, Neolithic cultures started to ferment grapes for wine making. On the same year, the Babylonians started to use microbial yeast for the production of beer. Second is Louis Pasteur. In 18,000s, Louis Pasteur has proven that the result of microbial activity was fermentation. And lastly is Sir Alexander Fleming. In 1928, Sir Alexander Fleming extracted penicillin that is made from mold, Penicillium notatum. Large-scale fermentation methods were developed in the 1940s to manufacture industrial amounts of this wonder drug. However, not until after World War II did the revolution in biotechnology begin, giving rise to modern industrial biotechnology. One of the mentioned people behind industrial biotechnology is Sir Alexander Fleming, and this is one of his sayings. You do not know what you will find. You may set out to find one thing and end up by discovering something entirely different. The people who are involved in industrial biotechnology are the engineers, scientists, analysts, and microbiologists. The engineers ensure the quality of the products for safety and efficacy, while the scientists are the ones who plan and perform experiments. On the other hand, the analysts evaluate how new products or technologies affect industry. Lastly, the microbiologists study microorganisms and develop products for industries. Finally, in this section, the people who benefited in industrial biotechnology are the industries and consumers. Industrial biotechnology saves resources and contribute to substantial reductions in greenhouse gas emissions, helping to mitigate global warming. For industries, it contributes to improved efficiency, sustainability, and higher value products.
The scope of industrial biotechnology, or white biotechnology, is quite grand, as it includes all of the industrial processes involving microorganisms. The biggest or the largest applications of this is in the food industry, as well as in the development and production of biofuels. Now, other applications also include the production of biodegradable polymers as well as industrially relevant enzymes. Now, we will be discussing three of the major ones, which is in food production, and biofuels, and polymer production. So let's take a look at the main tools or the key tools in white biotechnology, and these are biocatalysts. So we have several kinds of biocatalysts that are used, primarily including microbes such as anaerobic bacteria, example yeast. Lipases are enzymes that catalyze the hydrolysis of fats. Proteases, enzymes that break down proteins. Cellulases that break down cellulose, and amylases that break down starch into simpler sugars. Now, there are several advantages and drawbacks in using biocatalysts in comparison to chemical catalysts. So, first of all, we have the fact that it may operate at or near room temperature and pressure, whereas chemical catalysts usually require higher pressures and temperatures. Next, of course, is that it involves less complex equipment. However, both are expensive either way for both chemical-based catalysts and biotech or biocatalysts. Now, in terms of quality, that of course depends on the product. And then another good thing about it is that it contributes less to pollution because it is much more easily disposed of compared to chemical-based catalysts. However, there is certain difficulty in the practical application of biocatalysts in larger scales. For example, the development of bioplastics. While this does work in the small scale, there's quite an issue in large scale application. Now, so as you can see here, most of the biocatalysts utilized are actually enzymes. Enzymes are catalysts produced by living organisms to carry out specific biochemical reactions. Now, most of these enzymes are proteins. Each type of enzyme has a different shape, which fits the corresponding substrate only, and the place of connection is of course called the active site. Now, the active site is denatured or distorted in high temperatures and incorrect pH levels. Therefore, it is high maintenance, and enzymes, of course, are not alive. However, they are made by living organisms. Now let's look at the actual industrial applications. The use of enzymes is done at industrial level processing of food items, as well as enhancing its production. Food processing industries worldwide make use of the enzymes that are produced with help of organisms that are usually genetically modified. The enzymes thus produced include proteases and carbohydrates and all the other enzymes that I have previously mentioned. In order to get greater production in a smaller amount of time, cloning of the genes involved in the enzyme production is done. Now, these enzymes are used for various purposes, such as the production of cheese, making curd, and adding flavor to food items. Now, this industry actually utilizes 50% of all of the enzyme consumed by the industry. So let's look at some examples. First, we have catalase, which is used in mayonnaise production as it removes hydrogen peroxide. And then chymosin, useful in cheese production as it coagulates milk. We will be discussing this further in a little case study. Then we have glucose oxidase, which is used in baking as it stabilizes the dough. And then alpha amylase, which converts starch into maltose and which is used in baking or production of artificial sweeteners. And then protease is, of course, used for meat tenderizing or in baking and dairy products. Now, protease treatment is an efficient method for meat tenderization. Different proteases such as bromelain, papain, ficin, actinidin, and calpain are widely used in the proteolytic proteases, play an important role in degrading the structural proteins in the connective tissues and thus reducing the toughness of meat. And of course, the example we are all familiar with is the utilization of yeast in wine and beer production as they consume sugars, converting them into alcohol and carbon dioxide. Now let's look at a little case study on cheese production. Now, as you can see here, our focus on this would be in the coagulation or the curdling stage. As you can see here, fermenting microorganisms are added, as well as phenol, which is an enzyme. Now, rennet is commonly added to speed up the process of coagulation, although it of course still occurs naturally. Now, rennet is not one particular enzyme, but consists of multiple enzymes, which includes protease and pepsin, that are responsible for breaking down protein to form peptides and amino acids. Lipase, which converts fats into glycerol, and fatty acids also exists within the rennet. Now, renin and chymosin are enzymes that hold great significance in cheese production. Renin is responsible for curdling milk, while chymosin causes proteins in the milk to come together to form curds, and separate from the way known as glycomacropeptide by reacting with the substrate K-casein. Additionally, transformation of casein to paracasein under the influence of rennet and precipitation of paracasein in the presence of calcium ions also takes place during this process. So as you can see in the diagram, the coagulation part of this step is of course one of the most important steps in cheese making. So another important application of biotechnology is in the production of biofuels and bioenergy, or alternative fuel sources. Now, research in the production of these biofuels has been growing significantly over the past few years due to the rising energy prices and the depletion of fossil fuel reserves. Now, cellulosic biomass, in particular, offers an abundant and largely untapped resource for biofuels, if, of course, the optimal and most cost-effective conversion methodologies are determined. Biotechnology is being used to produce new advanced biofuels that perform more like gasoline in order to match the current performance of gasoline. Now, the scope of biotechnology spans feedstock chemistry, degradation processes, and variations in technology, of course. In the diagram, cellulosic biomass is particularly under the first generation category, where most of these biogas, bioethanol, biodiesel is produced via anaerobic digestion, some particularly utilizing the fermentation process, of course. And finally, we have the production of biopolymers. As we all know, during the last century, excessive usage of plastic products has caused a mounting concern for environmental and energy reasons, as inefficient waste management and inadequate recycling has resulted in the accumulation of plastics in the environment, which continues to significantly impact living organisms, both human and non human. Moreover, it is also heavily reliant on fossil fuels, which of course has its own environmental ramifications, as well as the cause for concern for declining supply and unstable markets. One of the main future oriented alternatives developed through industrial biotechnology is the production of polyhydroalkanoates, or PHAs. PHAs are polyesters produced by numerous prokaryotic strains from renewable resources like carbohydrates, lipids, alcohols, or organic acids, classically under unfavorable growth conditions due to imbalanced nutrient supply. Now, what are the main advantages? Why is the industry interested in this? First of all, of course, is biodegradability. This is a typical feature for biologically synthesized products to be again degraded by other living organisms. As such, PHAs do not contribute to an increase of the landfill mass due to their biodegradability, as they can be composted after use. Now, for the next advantage, we have its bio-based nature and independence from fossil fuels. PHAs are produced from renewable resources and, as such, are independent of the availability of fossil feedstocks. Now, this has a lot of potential considering if the energy it uses is generated from another renewable source, then it would be completely independent from the fossil fuel industry. Next, of course, we have is the carbon balance. If PHAs are applied instead of our regular fossil fuel derived polymers, the carbon cycle is closed. This is because to the fact that carbon sources used for biotechnological production derived from carbon dioxide, which was previously fixed by green plants. Hence, the release of CO2 by degradation of polyhydroalkanoates is just the final step of natural mineralization. So think about it like this. Plants that are capable of storing CO2 is then processed to make PHAs, and these PHAs, of course, will just release the CO2 back to the plants. Now, as we all know, CO2 is of course a problem as it is one of the greenhouse gases. And of course, this is an advantage because it will only be related to the amount originated from energy production and not to the carbon in the polymer itself. And then finally, we have biocompatibility. This is very important in specific fields of applications, particularly in medical applications where it may be used to prepare safe safe equipment or devices for, of course, the human body, such as implants and surgical devices, as they are superior to conventional plastics in biocompatibility.
Good day, I am Shaina Habanban and I am going to report on the following slides: the status of biotechnology in the Philippines. Starting on the policies and milestones. On December 20, 1979, the establishment of National Institute of Biotechnology and Applied Microbiology in UP Los Baños was approved during the 92nd meeting of the UP Board of Regents. And in the following year, National Treasury released an amount of 10 million out of the Energy Special Fund for the institute to fund the infrastructure of the building through a letter of instruction from President Marcos. So, the reason why the National Institute of Biotechnology and Applied Microbiology is established in the Philippines is that during 1970, the Philippines was going through an energy crisis and found out that Brazil is successful using alcogas, which is a sugarcane gasoline that can run their vehicles. Also, during this time, there is an increasing interest in biotechnology as a possibility of genetic engineering in the USA. That is why President Marcos instructed the Energy Minister Jeronimo T. Velasco and Chancellor Emil Q. Javier during the link between Brazil to find the link between Brazil and USA. Because of this, too, with the help of the groups they formed, they proposed on creating Institute of Biotechnology in the Philippines. So moving on, in March 1995, President Fidel V. Ramos issued Proclamation 526, constituting a network of National Institute of Biotechnology in the UP system, consisting of institute in UP Los Baños, UP Diliman, UP Manila, and UP Visayas. Biotech UPLB was renamed to National Institute of Molecular Biology and Biotechnology. As shown on the screen, it is presented here the timeline of the milestones in biotechnology from 1986 to 2002. During 1986 to 1992, President Corazon Aquino emphasizes on the Second ASEAN Science and Technology Week the emergence of new technologies, especially in the field of biotechnology. During this time, biotechnology in the Philippines is flourishing, starting with noteworthy accomplishments such as isolating a nitrogen fixing bacterium from native weed, or also known as talahib, that reduces nitrogen fertilizer input for rice and corn. Furthermore, in 1990, Executive Order 430 was created, which focuses on the duties and responsibilities of the National Committee on Biosafety of the Philippines. Some of the duties and responsibilities mentioned in this executive order is the ability of the mentioned National Committee to identify and evaluate potential hazards involved in initiating genetic engineering experiments and recommend measures to minimize the risks. From 1992 to 1998, Biotech Institutes is established in different University of the Philippines campus. And science technology programs gained more focus, which is under the presidency of Fidel V. Ramos. The year 1997 is the creation of the Republic Act number 8435, or also known as the Agriculture and Fisheries Modernization Act, which, as presented in the title of the act, focusing to modernize the agriculture and fisheries sector for the challenge brought by the globalization. On the act, it is also mentioned the partition of percentage where 40 or 4%, of the allocated budget will be used to support the biotechnology program. In 2000, the institutionalization of biotechnology in government program under President Estrada was established. Furthermore, President Arroyo gave a policy statement on modern biotechnology in 2001. Lastly, in 2002, the regulation for the importation and release into the environment of plants and plant products derived from the use of modern biotechnology has been established. This is to guarantee that the agricultural biotechnology products that are available in the market are safe. In this slide, it is presented the available industrial products in the Philippines as of November 2020, taken from Agricultural Biotechnology Annual Report. For plant biotech products such as BT eggplant, golden rice, BT cotton, delayed ripening papaya, genetic engineered corn, and biofertilizers are some of the products distributed in the country's market. On the other hand, there are no available genome-edited animals or clone reported in the country, while there is no information for the microbial biotechnology in the Philippines. One of the very important establishments that have been created is the Institute of Plant and Breeding, which focuses on strengthening plant breeding research in the country through the development of new and improved crop varieties. So it is shown on the screen the core functions of IBP, which is to assist other agencies in multiplying quality seeds and vegetative materials of recommended crop varieties, to collect, introduce, preserve, and maintain germplasm of important and potentially useful agricultural and horticultural crops, and lastly, to promote wide-scale use of IBP varieties and ensure that seeds are made available to small farmers. Institute of Plant Breeding is also the responsible institution for the development of the fruit and shoot borer resistant eggplant, or also known as BT eggplant. It is known that eggplant in the Philippines was cultivated on small family farms and is an important source of nutrition and cash income for many resource poor farmers. So the development of eggplant that can resist fruit and shoot borer, as shown in the figure at the left, is important because it is considered the most destructive insect pest for eggplant in Southeast Asia. To add up, IBP is also responsible for the development of the delayed ripening papaya with ring shot virus resistance. The first field of the test of for this project is already completed in 2014. Currently, IBP is organizing a team that will pursue the project and the possible second field of trial for this GM papaya. For this slide, I assume that everyone has heard about Golden Rice, which is a project of the Philippine Rice Research Institute or Field Rice. The main goal of the development of this GM rice is to address vitamin A deficiency, which is the main cause of blindness and death for malnourished children and pregnant mothers in the third world. Since the source of vitamin A is rarely part of the daily diet, the project aims to add beta carotene on rice, which is the dietary staple in the country. The two golden rice field tests were harvested in October 2019, and the Department of Agriculture's Bureau of Plant Industry or BPI approved the corresponding biosafety permit the following month, finding that golden rice is as safe as conventional rice in the market. Besides golden rice, other institutions perform research on projects such as BT cotton, which already obtained the certification of satisfactory completion on the multi-location test on November 11, 2018. Other plant biotech projects in the Philippines are herbicide-tolerant maize, virus-resistant sweet potato, and virus-resistant abaca.
Nung nag-establish itong biotech, 1980s pa, meron tayong energy crisis. Napakamahal ng gasolina. Ang gasolina, kinakailangan sa paggawa ng abono, yung mga inorganic. So, naisip namin, kaming mga researcher, pwede tayong gumawa ng fertilizer na buhay gamit ang mga mikrobyo, mga amat, mga bacteria. So, may isang buong program kami dito sa biotech na ang layunin ay mag-develop ng products na nagiging abono, biological fertilizer. So, ang mycoplast, siya yung pinakabunso ko sa ngayon. Kasi marami pang ginawa. Ang kagandahan sa mycorrhiza, so mycorrhiza yung common, yung general term. So, may pumapasok sa loob ng halaman. Nagsiserve sila as root extender.